there are 4 different aldolase B transcription factors. And transcription factors actually control the expression of genes in the liver, because aldolase B is found in the liver. Okay? What these transcription factors do is they control when, where, and how much of the DNA is read to be able to produce and express these genes to eventually produce protein. So, transcription factors interact with specific DNA sequences, and those specific DNA sequences they interact with are called DNA motifs. And they control whether a gene is turned on or off based on the metabolism that is happening at the particular time. Typically, transcription factors bind promoters, and promoters are found typically at the 5' end of the DNA. And the promoters will tell the RNA polymerase when to start the transcription process, and so that is how gene expression occurs. Now, there are 2 types of aldolase B transcription factors that I'm going to talk about with this slide, and they work in opposite concert with one another. The first one is HNF1, which is a hepatocyte nuclear factor 1. Like I said, HNF1. It deals with the expression of numerous liver genes that have to do with glucose metabolism and transport of glucose, including the expression to produce insulin, albumin, fibrinogen. But the HNF1 actually binds DNA as a homo or hetero binder. If it binds as a homo binder, that means it is alone when it forms the dimer. But also, the HNF1, there's actually an alpha and a beta. They can actually work as a team and bind together as well to the specific site on the DNA. So this actually will drive the expression of aldolase B transcription forward. The HNF1 does that. Now, to counteract that, there is an opposite, and the opposite is HNF3. And HNF3 stands for hematocyte nuclear factor 3. It actually controls, again, which genes are turned on and off and are allowed to be transcribed. HNF3 comes in 3 different forms. There's actually a alpha, beta, and gamma. But the gamma itself helps activate gluconeogenesis enzymes whenever blood sugar drops too low. That's a little side information. But HNF3 actually will compete competitively for the binding site on the DNA with HNF1. And so they actually fight for that same binding site. HNF1 drives the transcription forward. HNF3 stops the transcription. And so HNF1 is considered a superactivator, whereas HNF3 is considered a deactivator, and it stops the promotion of the aldolase B. Now, the concentration within the liver cells can actually determine whether HNF1 or HNF3 is active and is more likely to be transcribing genes for aldolase B, but it all depends on concentration. It's concentration based. There are also 2 other aldolase B transcription factors that I want to mention. The first one, and these 2 actually somewhat work in concert as well. The first one is, well, not really in concert, but they're similar to one another. The first one is CEBP, the second one is DBP. Now, both of these bind to the same spot. They both have a high affinity for the D binding site on the albumin promoter. And what this albumin promoter does is it can actually drive transcription. So the CEBP actually will drive transcription and help determine the number and the differentiation of different hepatic cells, activates certain types of promoters, and actually is there to promote steady state metabolism for the liver. The CEBP transcription factor keeps our metabolism pretty steady state and is present all throughout the day at all times during the day. And this transcription factor is present not only in adults but also in newborns. It has a classic leucine zipper structure, does form dimers, and keeps us at baseline levels. Okay. Now it is a weak aldolase B activator as well, when compared to the second transcription factor that I want to talk about, and that is DBP. DBP stands for the D-site binding protein. This one is found in adult liver tissue only, not in newborns, okay, adults only. It doesn't actually have the same leucine zipper structure as CEBP does, but this one is actually a strong transactivator for aldolase B. DBP actually will vary throughout the day. It follows some form of circadian rhythm. It's actually peaks its metabolism around the evening time. And then studies have shown that in the morning, the amount of DBP has actually gone down to an extremely low level. But one thing that's really interesting about DBP is it actually can block the HNF3. And it blocks it in a non-competitive fashion. They're not both fighting for the same active site on the DNA, technically. But the DBP can block the HNF3, which is really interesting as well. That then will promote aldolase B transcription. So aldolase B transcription really is promoted by HNF1 and by DBP. So in summary, glycolysis is the first step where we take glucose that we receive in our food, break it down, and through a series of steps following glycolysis, produce energy. Glycolysis is strictly an anaerobic pathway. No oxygen is needed. 2 ATP go in, and we get 4 ATP coming out, okay? In times of low glucose, though, we have to run a backup cycle. The backup cycle is necessary because if glucose levels are low in the brain, in cardiac tissue, etc., we need glucose to stay alive and to keep functioning with our metabolism. And so in that case, gluconeogenesis can be activated in the liver. Glycogen stores first can come out, and gluconeogenesis can also run side by side and work backward, bypassing those irreversible steps to produce glucose for the tissues that need glucose to function. Aldolase catalyzes to both a step in both glycolysis and gluconeogenesis, okay? Whereas 6-carbon goes to 2 3-carbon chains, or 2 3-carbon chains combine in gluconeogenesis to eventually form a glucose. So aldolase is very versatile in that fashion. Aldolase comes in 3 different flavors, depending upon their tissue, where they are found, as well as the speed with which they work. There is A, B, and C. Aldolase B was what we were focusing on for this presentation, found in the liver, okay? In the liver, that's where your gluconeogenesis is happening, as well as glycolysis. Aldolase B expression is controlled by different transcription factors. HNF1 promotes aldolase B transcription. HNF3 blocks transcription of aldolase B. And then there are other transcription factors as well that can either promote aldolase B transcription throughout the day. These are the references for this particular presentation. Thank you.